Good morning. Today we will do a practical on qualitative analysis of carbohydrates. The aim of this practical is to detect the presence or absence of carbohydrates in the given sample. Carbohydrates are organic compounds containing carbon, hydrogen and oxygen. They are classified into monosaccharides, oligosaccharides and polysaccharides. Today we will do some simple tests, also called as color test, to detect the presence of carbohydrates. The tests that we are going to do are Fehling's test, Iodine test, Benedict's test and Molisch test. We'll do the practicals now. I've taken a sample in which we have to detect the carbohydrates. Reagents: Fehling's A, Fehling's B, 10% iodine, Benedict's reagent, freshly prepared Molisch reagent and concentrated sulfuric acid. On one side I have taken a few clean glass test tubes, a spirit lamp, matchbox and a test tube holder.
So first we will do the Fehling's test. Take a clean dry test tube and add 2 ml of the sample. Then first add 2 ml of Fehling's A. Followed by 2 ml of Fehling's B. So we get a blue solution like this. Mix it thoroughly. We have to boil it. Take the spirit lamp. We will remove the reagents from here. And light it. Take the test tube holder and hold the test tube tightly. Gently start heating the solution. So you can see it is blue in color. Slightly keep moving the test tube. It has slowly started boiling. As the solution boils, we can see the color change. It first changes into green, then to orange, and then gives a sort of reddish brown precipitate. So this indicates the presence of carbohydrates. The second test we will do is iodine test. Take 2 ml of sample in a clean test tube. You can see here iodine is a sort of slightly brown, orangish brown in color. When you add it to the sample, if carbohydrates are present, the color of the sample changes into dark blue. This is a test for iodine. The third test that we are going to do is the Benedict's test. In a clean glass test tube, take about 2 ml of the sample. Add 2 3 ml of Benedict's reagent. So the color of the sample is blue, light blue. This also needed to be heated. Again take the spirit lamp. Light it. Hold the test tube with the test tube holder. So now you can see the solution is light blue in color. So let's see what will happen when we heat it. As we heat, we can see the color changes here also. It turns into green. So you can see the green color. Then if you keep heating it, it changes into orange and then into reddish brown.
The last test we will do today is the Molisch test. For this, we require Molisch reagent and concentrated H2SO4. In a clean test tube, take about 2 ml of sample. Add a few drops of Molisch reagent and mix well. Then add concentrated sulfuric acid along the sides of the test tube. So you have to be careful while using concentrated sulfuric acid. Hold the test tube in a slanting position and slowly add the acid along the sides of the test tube. So once you add sulfuric acid, you can see the formation of a purple ring at the junction of the two liquids, that is sulfuric acid and the analyte. Analyte meaning sample and Molisch reagent together is called as analyte. So between that and sulfuric acid, you can see a purple ring. So this indicates the presence of carbohydrates. Here I have kept all the test results. For Fehling's, we have got a reddish brown solution like this. For iodine test, we got a blue solution. For Benedict's, we got a orange or reddish orange precipitate. And for the Molisch test, here if you allow it to stand for some time, you can see clearly the acid and the analyte. In between that there is a purple color ring. So all these tests indicate the presence of carbohydrates in a given sample. Here I have given you the procedure of whatever tests we have done. This is how you have to write in the record: experiment, observation and inference, Fehling's test, iodine test, Benedict's test and Molisch test. After doing the test, you have to tabulate the results. This is a tabular column. On the left side, you have the 4 tests. Then how many of samples are given? Sometimes they might give you 2 samples for exam or 3 samples. So you have to write sample A, B, C and D. If they have given you 2 samples, then take 2 test tubes and label it as sample A and B and conduct all the 4 tests. If 3 samples are given, then you can take 3 test tubes, label it as A, B, C and conduct all the tests. The sample which I have taken today had both starch and glucose. That is why it answered for all the tests. Suppose if in a sample glucose is present and if starch is not there, it will not answer for iodine test. If starch is present and glucose is absent, it might not answer for Fehling's test and Benedict's test. And sometimes in exam they can give you a blank sample which will give you negative for all the tests. So it doesn't matter that or it is not necessary that it has to answer for all the 4 tests. Even if it answers for 1 or 2 tests, you can infer as carbohydrates are present in the sample. So after tabulating your results, you have to give the result as such and such sample contains carbohydrates and so sample does not have carbohydrates. You don't have to mention whether it is glucose or starch, just mention it as carbohydrates. Thank you.